Good afternoon, everyone. Firstly, I'd like to thank Anne Vakuliev, Don Lee, and Professor Chen for inviting me to present our work. I'm disappointed not to be able to join you in person this year in Shanghai and hope to see some of you at Escher in London in July. In this session titled The Mosaic Illusion, presumably referring to the continuing debate on the extent of chromosome mosaicism, I will be presenting some of our recent work combining microarray-based SNP parental haplotyping and intensity analysis to distinguish meiotic and mitotic aneuploidies in trophectoderm biopsies. This combination has enabled us to redefine intermediate or so-called mosaic copy number changes as mixtures of cells with normal biparental inheritance and cells with mitotic imbalances. This potentially has important clinical consequences as some of these embryos can then, with appropriate genetic counselling, be considered for transfer, minimising the discard of potentially viable embryos. So to begin with, I need to declare a potential conflict of interest. I'm founder of Exovo Genomics, which licenses software including Omnia and Anuscan for comprehensive pre-implantation genetic testing. We had access to a newly discovered DNA polymerase, which allows isothermal multiple displacement amplification, and began to test it on single and small numbers of cells. This was a turning point for pre-implantation genetic testing because we quickly realized that we could amplify the whole genome of these samples and obtain microgram quantities of DNA, that is to say, equivalent to Coryn-Willis sampling or amniocentesis. So at that point, we were very excited to begin to use microarrays, which were newly developed, for genotyping single nucleotide polymorphisms, or SNPs, which are very useful markers spread across the genome. And this eventually led to the publication in 2010 of a method to use these markers for parental haplotyping, or what we called at that time carrier mapping, which effectively provides a universal set of markers for linkage-based pre-implantation genetic testing of monogenic disease. Subsequently, in 2014, the method was commercialized and then validated by a number of labs around Europe and beyond. Compared to the requirement in the 1990s to develop disease and patient-specific tests for each couple, which often took months of work to develop and validate, this universal linkage-based approach has been revolutionary. And last year, one of the major testing companies, originally Genesis Genetics and now Cooper Genomics, published their experience in the 7 to 8 years after the method was validated and became available commercially. And I'm very grateful to Tony Gordon and Darren Griffin for collecting the data. So you can see that in this period of time, a staggering approximately 68,000 biopsy samples from over 14.5 thousand PGTM cycles were analyzed. This involved just over 9,000 couples at risk of conditions, including over 1,000 unique disorders, involving some 4,000 different mutations. But very importantly, in many cases, 73% of these samples were independently tested by sequencing and all confirmed to be accurate. Furthermore, over this period of time, the authors are not aware of any misdiagnoses, which is an amazing achievement. However, over this period, despite the fact that SNP arrays are well established in molecular cytogenetics for high-resolution copy number analysis, the SNP intensity information provided by the array was not used to test for aneuploidy. So in 2018, we began to develop a method for combining SNP parental haplotyping, which had been previously used for PGTM, alongside parental intensity analysis for the identification of meiotic and mitotic whole and segmental chromosome aneuploidies. And I'd like to acknowledge Jakub Horak and his team at GNT Labs in Prague, who encouraged and supported this work. So last year we published the results of using this approach to test a large number of trophectoderm biopsy samples in a series of mainly PGTM clinical cases. The study involved over 340 cycles of PGTM in couples with a mean maternal age of about 33. And remarkably, we were able to show that in this younger population of patients, less than half of the trophectoderm biopsy samples were in fact aneuploid. But more interestingly, we showed that an almost equal number of meiotic and mitotic aneuploidies, and mitotic aneuploidies with normal biparental inheritance, as we'll see, were detected. Furthermore, these aneuploidies included a high instance of segmental aneuploidies, mostly of mitotic origin. So this is what the plots of this combined analysis of the SNP data look like. Firstly, an algorithm is used that counts the number of haplotypes in successive 1 megabase regions of the paternal and maternal chromosomes without the requirement for a reference sample. This is important since analysis of even a single sample is possible. And secondly, parental intensity ratio is plotted at successive 5 megabase regions across each chromosome. So you can see the haplotype analysis in this ribbon across here, which shows the paternal and maternal chromosomes, and pale green indicates the normal inheritance of a single haplotype. So we're not actually displaying the haploblock regions of the parental chromosomes now, but just indicating that there's only a single haplotype present, and that's the normal situation. Down below, we show the parental intensity ratios, with the paternal intensity shown as blue dots and the maternal intensity ratio as red dots. And in these two normal samples, as demonstrated by the genotype analysis along the top, that shows normal single haplotypes for both the paternal and the maternal chromosomes, you can also see that the intensity ratios, the blue dots and the red dots for the paternal and maternal chromosomes, coincide, confirming that these both are euploid trophectoderm biopsy samples. On the other hand, if we now look at a sample from an abnormally fertilized embryo, in this case a digynic triploid, what you can see very prominently in terms of the haplotype analysis along the top is that for the lower maternal chromosome, you can see these purple regions in each of the chromosomes indicating abnormal fertilization. And if we zoom in on an individual chromosome, these dual haplotype regions are distal from the centromere, indicating M2 type trisomy, which is typical of digynic triploids because these are most often generated following ICSI and failure to extrude the second polar body. And equally then, if we look at the parental intensity ratios, that you can see that the maternal ratio is raised in comparison with the paternal ratio across all of the chromosomes. So this is a 69 XXX triploid, digynic triploid, and this is actually undetectable by NGS-based copy number analysis on its own. So this is a very powerful technique that correlates the two parameters, both the haplotype counting and the parental intensities. With meiotic aneuploidies, as illustrated in this sample with a maternal monosomy for chromosome 4, the absence of either haplotype for the maternal chromosome, shown in grey at the top, is confirmed by the maternal intensity ratio falling to baseline levels. And actually the paternal chromosome is present, but the paternal ratio is off scale here in this example. In contrast with mitotic aneuploidies, as illustrated in this sample by paternal gain of chromosome 1, both paternal and maternal chromosomes have a normal single haplotype, here demonstrated here in pale green, but the paternal intensity ratio is increased to the equivalent level as a meiotic trisomy. Thus, this mitotic imbalance has occurred in an embryo with normal biparental inheritance. Furthermore, using a reference sample, a normal haploblock pattern, which I haven't shown here, can actually be checked. Overall, in terms of the incidence per biopsy of these meiotic and mitotic whole and segmental chromosome aneuploidies, we were able to demonstrate an almost equal number, remarkably, of meiotic and mitotic errors. Meiotic aneuploidies were predominantly maternal and increased significantly with maternal age, as demonstrated here, as shown here, whereas mitotic imbalance was age independent. This has led us to suggest in a recent commentary in Reproductive Biomedicine Online that this additional information provides the option to consider how embryos are prioritized for transfer, with the potential benefit of minimizing the possible discard of viable embryos. And I'm just going to illustrate this here with two samples, one of which has a meiotic trisomy 16, which you can see here, and another sample which has two mitotic gains, one paternal and one maternal. So taking a closer look at the sample with the maternal meiotic trisomy for chromosome 16, you can see, and indeed aneuploidies of chromosome 16 do predominate in trophectoderm biopsy samples, and of course are a frequent cause of miscarriage. You can see clearly here when we analyze the haplotypes, you can see these dual haplotype regions either side of the centromere. So in this case, and indicated by the purple color, both on the P arms and the Q arms adjacent to the centromere. So this indicates an M1 type meiotic trisomy in contrast to the M2 type trisomies I showed you with the abnormally fertilized digonic triploid. This is correlated with an increase in the maternal intensity ratio, and conventional analysis also confirms this trisomy, since the AB band on the B allele frequency chart is split and there's a raised log R region. In contrast, if we now look at the high-level mitotic imbalances with normal biparental inheritance in the other sample, you can see here clearly from the haplotype analysis only a single paternal and maternal haplotype in both cases of these aneuploidies for chromosome 5 and chromosome 9. and correlated with the increased paternal intensity ratio here and the maternal intensity ratio here. Furthermore, again, conventional analysis shows this split AB band indicating gain, but of course it is what this allows us to do is actually say, well, this isn't a meiotic trisomy, this is actually a mitotic change. So conventional analysis doesn't actually differentiate of SNP intensity, does not differentiate between meiotic and mitotic aneuploidies. And just to show you one further interesting example, here we have a sample with multiple high and low level imbalance, mitotic imbalance, with essentially normal biparental inheritance. So you can see at the top here, the pale green across the whole of the genome of this sample. But down below you see chaotic changes in the parental intensities, sometimes with the maternal intensity increased, sometimes with the paternal intensity increase, and indeed you have high level imbalances and low level imbalances of the different chromosomes. So in this case, what we think is happening is that you've got a mixture of euploid and probably non-viable aneuploid cells, possibly resulting from late tripolar mitotic, a late mitotic tripolar mitotic division in the trophectoderm, as demonstrated recently by live cell imaging. So this could represent then, if this is just localized to the trophectoderm, this possibly indicates a euploid blastocyst with reduced implantation potential, because most of the cells resulting from tripolar divisions are non-viable because they're subdiploid, because the duplicated sets of chromosomes, instead of segregating to 2 cells, segregate to 3 cells. And live cell imaging has shown that these are maintained in the trophectoderm. So essentially you would have a whole area of the trophectoderm which would subsequently not be viable. So with all of this information available, we're basically suggesting that potentially it may be possible to change how we prioritized embryos for transfer, particularly single embryo transfers in PGTM and PGTA cases. And we can base this on known adverse clinical outcomes. So this pie chart here represents the proportion of euploid and meiotic aneuploidy that we had in the very large study of over 1,200 samples. But you can see here that quite a large proportion of samples actually only had mitotic abnormalities. So the options now available are perhaps even just to reject embryos with meiotic trisomies, because these can be very accurately identified using SNP arrays, and they're known to be the major cause of miscarriage. Furthermore, some of them are viable and result in abnormal fetal development and clinical syndromes. So you would certainly want to avoid transferring any sample that has a meiotic trisomy. On the other hand, if you just had a meiotic monosomy, as shown here for these samples, then potentially, because of the limitations of just sampling a small number of cells from the embryo, that apparent meiotic monosomy may in fact be mitotic and not affect the whole embryo. And in any case, if it is representative of the embryo, most of those are not viable and are lost very early on. Either they don't implant or they're lost soon after implantation. So again, with proper genetic counselling, they could be considered for transfer. And finally, depending on whether these samples with just mitotic errors could be considered for transfer, depending on the chromosomes involved, whether they're whole chromosomes or segmental aneuploidies, and so on. But some of these then potentially could be considered with proper counselling as transferable. So in that way, I would hope that this more complete picture of aneuploidies at these early stages in the trophectoderm samples from blastocyst stage embryos, we can minimize the discard of potentially viable embryos. So to summarize, this combined SNP parental haplotyping and intensity analysis has redefined chromosome mosaicism. It can identify meiotic aneuploidies, but redefines trophectoderm biopsy samples previously classified as mosaic by copy number analysis alone as having mitotic imbalance against a background of normal biparental inheritance. In other words, it identifies the fact that there are normal cells and a complete normal set of chromosomes from both the father and the mother. So with appropriate genetic counselling, and depending on the chromosomes involved and whether it's a whole chromosome involved or a segment of a chromosome, these embryos can potentially be considered for transfer. As I've already said, recent live cell imaging studies, beautiful studies by these two groups, has revealed that mitotic aneuploidies continue to arise both through abnormal mitosis and therefore affecting the whole genome, as well as affecting segregation of individual chromosomes, continue to arise in the trophectoderm at the blastocyst stage. And indeed, if you follow those cells through the subsequent divisions, the abnormal cells remain mostly in the trophectoderm. So creating patches of cells that are picked up as abnormal. So it's vital, I think, that we're able to distinguish these different types of aneuploidies to give the patient a good indication of what is wrong with their embryo, and perhaps to prioritize other uniformly normal euploid embryos. And indeed, the identification of complex combinations of mitotic imbalances, which is not uncommon, may indicate these abnormal mitoses that I've just described in the trophectoderm, and may be useful to identify blastocysts with lower potential for implantation. So there's a lot more clinical work that needs to be done, and I'm looking forward to collaborating with groups to look at the clinical consequences of these different types of abnormalities. Finally, I'd like to acknowledge my friends and colleagues who have collaborated in this research in various labs across the world, in fact. But particularly Jakob Horak, as I've already said, at GNT Labs in Prague, Tristan Hardy and Emily Button at Repromed in Adelaide, Australia, and Carles Jimenez, Maria Sandalinas, and Estefania Toro at Dina Science in Barcelona, and of course my colleagues at Exova Genomics, Matt Newnham, who has been instrumental in developing the cloud-based application which we use, and Alan Thornhill. So thank you very much for your attention. Thank you.